right, so thank you very much to everyone for coming. It's a really great pleasure for me to welcome Dr. Pilou Bazin, Pilou. I've seemingly known Pilou for a big chunk of my academic career, and we've had a lot of intersections in terms of the topics we worked on along the way, so it's a particular joy for me to be able to host him today. Dr. Bazin is a senior researcher in the Integrative Model-Based Cognitive Neuroscience, IMCN laboratory of the Psychology Department at the University of Amsterdam. And he also works at the Max Planck Institute for Human Cognitive and Brain Science in Leipzig, Germany. His interests lie at the intersection between cognitive neuroscience, magnetic resonance imaging, and medical imaging computing, with a particular focus on computational neuroanatomy using advanced MRI techniques. In addition to their implication, using these techniques as a way of looking at the implications on brain structure for cognitive function and human behavior. So with that, I'll thank you very much, Pilou, for accepting the invite, and I'll turn over the Zoom presentation to you.
Thank you very much for having me. It's always so yeah, I'm very excited actually to present this work to you guys, because you know, we've been doing a lot of works in similar directions, looking at especially this kind of small bits in the middle of the brain that people tend to just throw away and not look at, the subcortex. So I'm gonna try to show you a little bit some of our recent work, both on the technical side, trying to push a bit the method that we use to look at the subcortex, and also some recent results we got on aging in the lifespan. If there's question at any point, because I will switch topics quite a lot, don't hesitate to just interrupt me and ask. So a starting point, there's a kind of a funny quote from A.D. Johnson-Berg from now almost 10 years, geez, that one curious features of many cognitive neuroscience studies is that they're all about the cortex, completely ignoring subcortical or cerebellar structures. And her point that I find very interesting here is that she sees it more as an accident than a deliberate strategy. So why did we focus on the cortex? Well, first, I mean, it's traditionally the region that has been most popular, from the Brodmann work and the early neuroanatomy, all the way to some of the fanciest results we've had today that are really just looking at things on the cortical surface. But if you start looking in the other direction, it is a bit odd, because the subcortex, if you start counting the number of structures, for instance, is also vastly complex and vastly structured in a lot of different regions with different anatomy, with different composition, and therefore very likely some different function as well. And you can see a lot of them even with MRI, although, as I will detail a bit more, we need to zoom quite a lot. Another thing too that would bring us to look at the subcortex a bit more in detail is that it's a region of very strong interest in treatment of symptoms in severe cases of movement disorders, such as Parkinson's disease, OCD, anorexia, depression, pain, cluster headache, epilepsy, cases where people will undergo surgery to get deep brain stimulation electrodes implanted, not in the cortex, in deep regions of the subcortex. Another paper that I stumbled on actually a few years back was a very interesting study looking at the genetics of Neanderthal in us. So we all have Neanderthal genes, but thankfully for us, they are very downregulated. And in that study, they showed that they were actually very downregulated in brain regions, here represented in blue in that diagram. But what I found very interesting there is that if you look at the labels of the different brain regions that are most unregulated, we start with caudate, amygdala, cerebellum, spinal cord, putamen, nucleus accumbens, hippocampus. And at some point, we start looking at the cerebral cortex. So it seems that those small regions that we don't care about maybe actually are very important in making us more human and less Neanderthal. Likewise, looking at evolution in a maybe slightly wider scale, I found the work of Susanna Herculano-Hausel quite interesting, showing in particular that if you count the number of neurons and you see the interactions between cerebrum and cerebellum, even though it seems that we have very big cerebra, our cerebellum is actually not that small. And in terms of neurons, the relationships between cerebral and cerebellar neurons is very stable across species. And even though we have big frontal lobes and big cortices in general, our very small cerebella are very, very packed with a lot of neurons. So I hope with that I can convince you that it's a good time to start looking beyond the surface view. So rather than just focusing on the flow of information from cortical to cortical area, which is very interesting and has been a strong focus on neuroscience, and therefore we actually know a lot about this. It's a good time now to start remembering the kind of old textbook circuits of neuroscience, which involves a lot of loops between cortical and subcortical structures, just passing information back and forth in those different systems. And indeed, it seems that the subcortex has some important features that are important evolutionary. Places like the cerebellum are also fairly complex because they still have the majority of neurons. They must be doing something interesting. And last and not least, for clinical applications, it seems those regions are very important for things like deep brain stimulation, but also to look at early neurodegeneration in disease like Alzheimer's disease, where, for instance, the locus coeruleus recently has been very implicated into very early activities, dysregulation. So why not looking at the subcortex? Why have we not been doing it? Well, it's a very challenging area. It's challenging for many different reasons, and it's challenging for both anatomical and functional imaging. We've done some work on the functional side that I will not cover now because that's kind of another talk, and we're only starting to really get some interesting positive results there. But on the anatomical side, so the first thing that's fairly obvious is the subcortical regions are just small. So if you have classical resolution 3 Tesla images, even at 1 millimeter, you don't see a lot. They really start to come into focus in all their richness in postmortem MRI and even more in microscopy. The second part that is tricky is the fact that they're not only small, but they're all packed together. So it's very easy to miss one or to mislabel some, or to start looking at clusters that are actually very heterogeneous. And the last part that is, I think, interesting for the MRI scientists as well, is that they do require multiple contrasts. They have different compositions, especially in terms of iron content. And so looking at the different structures requires you to acquire different type of images together. So, well, we can maybe try to get an MRI scanner, if possible, 7 Tesla or above, crank up the machine, get the high resolution. So here is what I find still the highest record in terms of resolution with quantitative MRI. So 350 micron isotropic voxels. Here we're looking at T1 map. And you can see in those little regions, there is a lot of details. You can see some beautiful subregions of the hippocampus, some of the stria of Genari, and in the subcortex, a lot of details of the subcortical anatomy. However, this takes completely unpractical amounts of time. So we're looking at 4 sessions of 31 minutes each. So that's 2 hours for just the T1 map. So we've been working on trying to find some more practical approach. So in our lab, we've been working with Matan Khan and Witzke van der Svaar in Amsterdam to build the multi-echo version of the MP2 range. So MP2 range me. And here what happens is we're acquiring at the same time a multi-echo image for T2 star and QSM and a multi-inversion image for the T1 mapping. And the nice thing is we can do that together interleaved. So the T1, T2 star and QSM are inherently registered because the data is acquired at the same time. It takes about 18 minutes at 0.7 millimeter. And the nice thing also is that there's very little dead time. So you don't spend a lot of time waiting, you just acquire data almost continuously. So we get 10 images corresponding to the first inversion and the second inversion for echoes with both magnitude and phase. And then we can use a combination of those images. And it's also nice that we use all the data in that sense to obtain the quantitative T1, T2 star and susceptibility maps. One thing that is also interesting is even with those 6% dead time, we had still some space to put fat navigators. So those were particularly prominent in the work of Dan Galishan on the 350 micron images, which were useful there to correct for motion. So what we do here, we get a very fast image of the fat content of the head, which allows us to find a orientation and translation parameters for each slice that is acquired sequentially. And then we can reconstruct the images not using the classical FFT grid, but using a non-uniform version based on those displaced regions. And then we can correct the data. And you can see in those examples that we gain a lot of sharpness, we get a lot of precision in some of the structures, and some regions might also appear where they were not clear there. So we try to quantify this in a very, very practical, very neuroscience approach, which was, well, how much sharpness do we gain by doing the correction? So we looked at the boundary of different structures, the ventricles, the thalamus, the striatum, and the gray and white matter boundary. And then there we looked at the sharpness of the transition between structures. And you can see, so if I do it globally here on the bottom, you can see without correction how the sharpness is a very, very clear linear function of the displacement. Even for the small displacement, you can see a very strong relationship, whereas once corrected, it almost all the time goes back to the baseline resolution of your data. Another thing that was interesting in that study was that when we looked separately at those different regions, we could also see that some different structures have also different inherent sharpness. You can see, for instance, that the gray-white boundary, which is actually fairly smooth when you look at microscopy, is indeed less sharp than regions, that boundaries of the ventricle or the striatum, for instance. Okay, so we have those nice images. They are sharp because there's not motion, but they still have a lot of noise because we want to go fast and we want to get high resolution. So our SNR is not great. So how can we smooth them? Sorry, how can we remove that noise, but not smooth them? Smoothing would be an obvious solution. The problem is that you're losing resolution while you do that. So why acquire the high resolution in the first place? So there was a very nice work, actually from Montreal at the time, from Mangeon and Coupé, on diffusion imaging. That is now actually one of the standard steps in pipelines like MR-Trix, which was to use a PCA approach to denoise not in space, but in terms of the different directions that have been acquired. So if you look at a patch of the data and then compare across the different images that have been acquired, you should have some variation that are similar, at least in some of those images, because they're part of the structure. And you have some that just change all the time, and those are noise. And so finding the boundary between the two, you can then separate the two, hopefully. The tricky part was that in their case, they have about 60 or more images, typically from diffusion data, all with the same style or magnitude, whereas here we only have 10 images with magnitude and phase. And so we had to adapt a few things. The first thing we had to do was to take the phase and unwarp it, and then use both the magnitude and phase together to create a complex image. The advantage of that is both images have the same scales and the same noise patterns. And then the second one was to build a more practical thresholding technique compared to the random matrix theory to deal with being able to make a decision even with those 10 eigenvalues. So if you have a very good SNR in this region here, you can see that both methods work fairly similarly. So you have here in this case, we would have the perfect cut here, and the random matrix theory actually does it better in this case. But interestingly, when you start, for instance, interpolating, random matrix theory has a lot of problem with that. So that can be a problem in some cases. One thing that's also interesting is, of course, when you have low SNR, then it becomes a lot harder to get those separation. But in all cases, the threshold that we have tend to be more conservative, so keeping a bit of noise in rather than removing data. So this is an example. On the top, you have the singular values corresponding to the different amount of signal that you have in the patches. And in the part below, you have the eigenvectors. And it's very clear at the beginning that we have a lot of structure, a lot of anatomy in them. And at the end, you're very much in just buried in noise. And then when you reconstruct after removing those noise components, you can appreciate that the images have been very much cleaned up, but a lot of the very small details, things like the vasculature or the boundaries of the different structures have been maintained. We've quantified a little bit the improvements in some of the subcortical regions, and we can see something from about 5 to 20–30% fairly consistently And it depends also on the type of images you're looking at. For instance, with QSM, we had very little improvement because QSM already includes some spatial regularization. So now that we have those denoised images, we also wanted to have a slightly better representation of the subcortex. So here, that's segmentation we had previously, which is already pretty nice, but we wanted to see if we can increase on that. And that's the last big step before the application was to create a new method for segmenting a lot of the structures, including the small ones, in a way that is fairly concise. So we've used a Bayesian approach, trying to build sets of priors, looking at location, looking at skeletons, so the location of the inner part of the structures, and looking at the quantitative MRI contrasts. And then from that, building posteriors on the voxels, looking at Markov random field regularization, and then growing the structure backs. With that, we could segment 17 structures, here shown in a kind of exploded version on the top. And we could compare how we did in terms of test-retests reliability between human raters and our techniques. So now that we've have kind of all the technology in place, we've been also assembling at the same time a large database large for 7 Tesla, so only 105 subjects, but for 70 study, it's still quite challenging. And we use the MP2REGME sequence to obtain T1, T2 star, and QSM measurements. We try to get a sample that is as balanced as possible in each of the different age groups. So for each decade, we had about 13 or 12 to 13 subjects, with the exception of the youngest group, for which we had a lot more psychology students. So looking at this database, we wanted to see what were the effects of aging in all the different subcortical structures that we identified. So we know from previous works that there should be some active myelination and demyelination during the adult lifespan, some iron accumulation, atrophy of the nuclei, and also some local shifts that were something discovered by Max Planck nearly 10 years ago now. That was particularly important for things like deep brain stimulation, because if your nuclei start moving, your atlas is not as accurate as you think. So in order to look at iron and myelin, we went one step further than just looking at the quantitative MRI measurements. And we were inspired by the work of Carsten Stuber back in the Leipzig group with Robert Turner, that described a linear relationships between iron, myelin, and the R1, R2 star, and QSM parameters. There was a follow-up work from Riccardo Mettere, also in Leipzig, that was very interesting, where he found some fairly old literature that had been measuring myelin and iron concentrations in various regions of the cortex. Oh, I'm sorry, cortex of the brain, and could therefore give us some calibration study values that we could use with the in vivo measurements. And because those measurements were fairly sparse, we also used some data from another project looking at ex vivo data with a post-mortem study where we had full brain stains of the of silver looking at myelin, where we could then compare relative concentrations between regions. So putting all of these things together, we define relationships between those 5 components. You can see here on the planes, the winning relationship. We tried a lot of different options. So we did a model comparison with a Bayesian model criterion. And it was fairly clear that we have winning models that iron is well described with a mixture of R2 star and QSM, while myelin is described better with a mixture of R1 and R2 star. So that also corresponds to what we knew about those contrasts, which is quite nice. And when we then take the values from our data and compare them to what we had from the literature or from the relative stain measurements, we can see that the population averages are well matched in terms of the concentration of iron and myelin that we obtain.
If I could ask a question. This work is really lovely, and it's such an innovative way to get at kind of the underlying architecture of the signal that you're working in. I'm wondering, you talked a bit about generalizability, but if you take those kind of solutions and say you want to transpose it down into 3T or something like that, what would your, because that's probably where people have maybe a bit more data, but not with all of the, the other advantages you get from 7T, which also have their challenges too. So can you maybe talk a little bit about that, like the generalizability more grossly, or do you feel like this is more of a, something that can be used, but in a specific context? If you have any thoughts on that.
Yeah, no, that's actually a very good point, because here what we do, so if we were just looking at the quantitative MRI contrasts, then it becomes very challenging, because if you just have different contrasts, then you cannot really compare, you cannot just bring everything together, you cannot aggregate data. But because we have that extra step of calibrating to literature values, you could try to do the same thing with different sets of contrasts or different sequences. So for instance, if you don't have MP2 range means, but instead you work with MPMs, you also get the quantitative MRI contrast, but they're not exactly the same. If you do the R1, the B1 correction on R1 estimates or not, you also have slightly different values. But in the end, if you then bring that to the linear model, then you should end up with myelin and iron map that, even though the coefficients that relate them to the parameters, to the MRI measurement that you have are different, should also give you something that fits those parameters. So my hope here is actually that with contrasts that are rich enough, which can be difficult to get sometimes, but if you have enough contrast that shows you some myelin and some iron dependencies, you can build those linear and maybe even nonlinear model in some cases that correspond to those literature calibrated values. And then those images that have been recalibrated to the same scale are basically all comparable.
Right. And so would you then need a local kind of quantitative microscopy measure as well to kind of get to that, or?
No, so here, especially if you really want to compare across the different studies, you can actually use, so those values that we have here, those have been obtained from those early papers, so from the 30s and from 58, I think. So fairly ancient measurements. I think we actually should get better measurements now and maybe reactualize and get more measurements more precisely obtained. But if that's what we have, you know, that's kind of the standard that we go to. And when we create those models, of course, then when you have different data, those parameters should be different. But you still get models that end up giving you values that should be in the same range. And so if your data has been calibrated, then it's within the same spacing. And then the differences that you obtain should be comparable across sites.
Great, thank you so much.
And so here's a few examples. I like quite nicely how it shows nicely how the globus pallidus, which is often seen as white matter in the T1 weighted images, becomes a lot more like a classical gray matter structure, which it is, when you pull out the iron components and just look at the myelin. So we are almost there with the methods. There was one last thing we wanted to add, that when we look at morphometry, we often do things in terms of volumes, which is a global measure, which can be very noisy, especially when you look at very small structures. But when you think about what happens in the cortex, people have been very successful using cortical thickness instead, which is a local measure, which has a lot more interesting properties that way. So how can we bring that to subcortex? So using medial shape approaches, you can build a sign distance function that will create a kind of a little hill within your structure. And then you take the ridge of that structure, is a skeleton of it inside. And then you compute again the distance from that skeleton back to your boundary. And with that, you obtain basically a sort of thickness measurement, an equivalent to the cortical one, that you can use in any kind of structure with any shape. So here's some examples that we have from the lateral ventricles, where we create a skeleton, and then you can get the local thickness measurements. And that idea actually is not new. We also found that Boris Gutman and colleagues have been using it for quite a while now, and we also show recently some nice results in subcortical regions with it. So now that we have everything together, let's see if we find any interesting changes. So looking at myelination, iron, atrophy, and also local shifts. So first, let's look at myelination. It's a very busy slide. I'm sorry, those results are fairly complex. So here you can see for all the different structures, an example, an average of the young structure and the old structure, the color corresponding to the intensity of the mean intensity of the here given by the median, and some speckled based on the intensity variance is represented by the interquartile range. So you can see a lot of those different regions. You can notice also in some cases there's some separation between male and female, for which we have two different curves, but whenever we have one curve, it means a model that look at them together was the winning model. So we computed different models looking at node change, linear change, quadratic change, and so on. And then again, did a model selection to find which one was best explaining our data. Overall, looking at myelination, you can see that several of the structures have decreased in myelination. So it's STN, striatum, thalamus, red nucleus, fornix, all go down. And we also find fairly widespread an increase in the variability, so increase in the heterogeneity of myelination in all these structures. However, you can also see that some structures, for instance, even the amygdala, which we would not have expected, can be fairly stable. When we look at iron with the same representation, you can see also a slightly different structure picture, because you have structures that are iron rich as a baseline, so the globus pallidus, red nucleus, substantia nigra, STN. And those will tend to have more iron dynamics. So often with an increase, sometimes with an increase and possibly a decrease at the end. And also a fairly strong increase in heterogeneity. Another thing to note is that the striatum, even though it's lower as a baseline, tends to also have an increase in iron and a strong increase in the heterogeneity as well. Same thing, for instance, for the VTA. Other regions, so for instance, the PAG and PPN deep in the brainstem are very stable. And finally, when we look at the atrophy, so here we've been looking first at the median and and the interquartile range of the thickness. And we also looked at the volume just to be complete. And now we also have the ventricles. So with the ventricles, as you would expect, you have an increase in size of the lateral ventricles and the third ventricle, not so much on the fourth. And you can see atrophy in places like the striatum, the thalamus, the amygdala, the globus pallidus, pars externa, and so on. And also some slight increase also in the heterogeneity in some regions, and a decrease of the heterogeneity in other regions. So here the heterogeneity of the thickness would be meaning that the shape is more complex, whereas a structure that has low heterogeneity would be more like a sphere. So you can see it, for instance, with a red nucleus. That would be a typically fairly boring structure in that sense. The last thing we did was also to look at the local shifts. So that was quite surprising. We were expecting shifts sideways with the expansion of the ventricles. But if you look at the different structures, most of the shifts are more a settling down in terms of going lower down in the axial plane, meaning that with the atrophy of the brain in general, with the brain structure tend to sink a little bit rather than just spread out. So of course, all of this is a very complicated picture, so we try to make it a little simpler by doing spider plots and trying to find fingerprints. We can see that there are some patterns that are a little shared. For instance, the striatum, the STN, and the GPE have sort of the same approach, of the RN as well, red nucleus. But there's quite a lot of structures that have also their own pattern that is very different from the rest. For instance, the amygdala or the claustrum just have their own thing going on. In order to really examine those results and puzzle them a little further, we developed an app to look at them online, so we can look at the structure in 3D. We can rotate them, we can remove some of them, so we can just look at the ones we're interested in. We can pull the different curves that have been obtained for the different measurements and structures, we can also pull out the data so if we want to do reanalysis or combine it with something else. So that is the end of my talk. I hope I conveyed the impression that subcortical structures are important, but I think you guys knew that already. I would agree that when you have the opportunity using multimodal MRI, ultra-field, if you can, it really helps in terms of getting the resolution and getting the specificity of the signal. And the patterns we could observe on that first data set that we looked at show that it's indeed a fairly complex set of structures with a lot of different things that happen with different time frame. Last but not least, of course, that's the work of many people. So this is the IMCN lab. And I want to highlight in particular people who've been really involved in this study. Matan Khan has been really helping a lot with all the MRI side of things. Max Kirken has been doing a lot of the previous studies that we've been basing the work on and helped also with this. Anneke Alkemade has been doing a lot of the anatomy and delineations of the different regions we're interested in. Birte Vossmann has been driving the entire enterprise and keeping us on track. And Stefan Militic in particular, I'm very thankful for him because he took a lot of the kind of vague ideas I had about how to look at the different age differences and put everything into a very clean statistical framework and putting very, very clear results out from a very complex problem. Last slide, I just want to emphasize also that we really, really like open science, and we don't really like just using it. We want to also make it happen. So we released the entire database that the study is based on, the trajectory viewer that we built, and all the tools that I described are integrated into the NIRIS toolbox. And with this, I'm done, and I hope you have a few questions.
Bravo, thanks so much. What a wonderful talk. There's a lot of time for questions, so if anyone has anything, please raise your hand or actually put stuff in the chat and I'll transcribe it vocally, I guess. Aurélie, you're first up.
Yes, hi, thank you very much for the talk. That was very interesting. I had a question regarding the way you did your modeling to extract the iron and myelin maps. How do you think it will be done in the context of Alzheimer's disease, for example? Like, do you think the way you model could be applicable to a population with Alzheimer's disease participants, or do you think the modeling should be reframed?
Yeah, so it's a bit of a tricky question because the modeling is based on calibrating to those classical measurements. So those have been obtained on, in the most part, healthy people. Sometimes it's not very clear from the paper that sometimes there was a bit of a mix. So of course, then you want to calibrate on healthy population. So if you have Alzheimer's disease patients and healthy controls, you would use the healthy controls to build the relationship. And then you can use it to transform the images from Alzheimer's disease patients. We have, for instance, some Parkinson's disease patients. And so we're using the same modeling there as well.
Okay, so you don't think, for example, the amyloid plaques could interfere in the prediction of the content?
Well, they will give you different numbers. Then interpreting them as myelin or iron changes is it's a choice. Of course, the models that we build, we know they're not good in that sense. We know that R1 and R2 star are measuring a lot of different things. There's calcium involved, there's copper involvement in some places. So those models are definitely very crude, but they're starting points, and they allow us to talk about those two things separately, and also to have a calibration mechanisms that bring different type of images together.
At that point, do you need, because all of those are ROI level indices, right? They're not, how confident would you be about moving that to some version of a voxel-wise measure, for example? Or would you have any thoughts about how that transition would take place?
Yeah, so with that, the linear relationship is very broad. So you're basically just projecting your free MRI indices into 2 dimensions.
Yeah.
And whatever noise or variation of the signal that you have in the original data will be there in the transformed data. And so when you look at the images that we have, they actually have a lot of detail in them and a lot of sub-regional variation that can be used, definitely.
Yeah, no, that's exciting. Also back to Orly's question, I mean, would one possibility there be, you would end up having to have your normative model, and then if you have your 2 other groups, you would then interpret those results based on what you see in the normative model, right? Is that kind of what I got out of your answer, or is that a possible way? Like, you wouldn't want to model it to the pathological conditions. You'd want to keep everything kind of relative to that, right? And see how far you move away from that.
Yeah, exactly.
Yeah, okay. Yeah, Julia.
That was a very great talk. I have 2 technical questions because I'm not sure, maybe I missed some details. So one was about your denoising strategy and one was about the parcellation. Could you just repeat? So the denoising that you went through, that was based on the multi-echo MP2 range sequence?
Yes.
Okay. So my question is, what other, because I'm not familiar with multi-echo structural, we do multi-echo functioning in the lab, and we have, multiple metrics to assess the quality of our denoising. And you showed us one, which is the improvement in SNR. I was just wondering if it's, common practice to show other metrics, or was that the only metric you used to assess the quality of the denoising?
Yeah, so for that we looked at SNR at the time. We did the study looking at sharpness of the boundaries afterwards, and I think that would have been more interesting to add there. I mean, looking at SNR is a quick way to quantify how much change we get. But I think if you have an endpoint, so in the case of structural data, how well you can delineate a structure of interest, then you're in much better ground to see, well, this method is actually useful or it's not really changing anything. Yeah.
Okay, thank you. And then my second question was on the parcellation. Again, I'm not sure I fully got that part. So you said you used a Bayesian algorithm, right, to develop the parcellation?
Yes.
Is that done on the whole group? So how exactly was that carried out?
No, so we've used a practical approach that our neuroanatomists refused to do delineations of 105 subjects for all the different structures for some reason. So what we did is we used 10 subjects that were used as an atlas, and so we learn the statistical priors and things like that on those 10 subjects. I've done experiments looking at the number of subjects that you need to have a stable result. It's something between 3 and 5 typically. Of course, it means that we also have some biases in terms of how the different age groups could be represented. And those can be a problem, especially when you start looking more at the volumes. So we tested on a few of the structures, volume differences in more aged groups. But if you look at things like the quantitative MRI measurements, those are extremely stable, regardless of the precision of the delineation.
Okay. Yeah, because I guess my question here was, given also what you showed us with age, to what extent would we benefit from, an individualized parcellation approach that we have in functional data, also for us, the subcortex, given, all those changes that you showed us as a function of age?
Yeah, so it depends on your fMRI resolution also, of course. So a lot of those changes that we've seen in terms of location, for instance, that would be probably the more important ones. They're not very big in terms of the scale. So we're looking at a few millimeters. Of course, if you're doing deep brain stimulation and trying to implant an electrode on the tip of the STN, a few millimeters is a huge size, a huge difference. But if you're looking at 3 millimeter voxels, that's not so much of a concern in that sense. So it really depends.
Okay, thank you so much.
All right, thanks, Julia. Evelyn? Nice.
Hey, yeah, sorry, thanks. That was really, really cool. So I'm coming to this from a functional perspective in preclinical imaging, so and definitely out of my depth in a lot of ways. But I found the differences between regions and the heterogeneity that you were seeing correlated with like either myelin in different regions was really interesting. Have you tried to sort of connect that back to any other information you have on those individuals? Like is the heterogeneity correlated with any interesting metric you have on those individuals, or is that somewhere you're going next? Just sort of expand on that point.
Yeah. So we've been a bit, I guess, a bit naive at collection time. We decided to create just an imaging database in that sense, and so we don't have additional measurements. We don't have tests. We don't have questionnaires to help us with. On the other hand, you still have to remember that we're just looking at 100 subjects over the lifespan. So making big generalization becomes really hard because your sample size is very small still. So that's why we're interested to see how much we can then bring that kind of calibrated view to other databases, because if you have a few thousand people, then you can start pulling those factors apart, definitely. I agree that one thing that was very puzzling is that the heterogeneity of the system definitely increases with age. So everything becomes less similar, and it makes sense. You're aging, you're changing, and you have more of a life path that's behind you. That's also part of you. So, but it makes things more challenging because you should have more data and more participants in order to be able to say anything there.
Very cool. Thanks.
Fantastic. Are there any other questions? Anyways, I'm very grateful to Pilou for this wonderful talk. So, and thank you all for your attention. If I know, Pilou, you have a hard stop in about a half an hour, but if there's anyone who'd like to stick around for the for student discussion or trainee discussion, please do, and I will vacate the premises so that I won't pollute the environment. So thank you so much. Wonderful talk, and thank you all for